We are now live because we just had the presentation of Cassandre, was still around in the background. Cassandre from the Philippines, the presentation was not online. And so I just tell everyone online that our jury members are Aziz Moula, Steve Mo, Nadir Lafayette, and Anne Pajot. Isaac, are you all good?
Will be, yes.
Just before Isaac starts, just to remind you and those arriving that when we are live like that, we have a network of microphones that can pick your discussions whispers much more than what we can hear in the room. So try to not comment on the presentations when it's live. Isaac, your turn.
Okay, so hello everyone. I'm Isaac and I'm here on behalf of Nacho Dive, who's very present. Thank you. And I'm going to be talking about applying citizen science to survey the ecological status of the marine environment. So for centuries, marine biodiversity has been linked to socioeconomic well-being in Mediterranean coastal zones. Threats to the health of marine biodiversity directly impact ecosystem services, thus falling disproportionately on human populations that rely on them. Among these threats are poor fishing practices and overexploitation by both small-scale and recreational fisheries, which have led to a slow degradation of marine biodiversity and the conservation status of many pelagic fish species. In fact, more than 8% of Mediterranean fishes are listed as vulnerable, endangered, or critically endangered as of an IUCN report. One of the most recognized conservation tools are MPAs, or marine protected areas, specifically designed for enhancing marine biodiversity and safeguarding ecosystem services. Under this protection umbrella are no-take zones, which have been specifically applied to manage overexploitation by fisheries. With more than 70% of MPAs lacking basic management, this really hinders their real ecological benefits and highlights the need for efficient monitoring programs. But monitoring fish communities or monitoring fish assemblages, referring to fish communities in a given area, can be difficult as fish fulfill a wide range of ecological roles in an ecosystem and inhabit a variety of different habitats. The most productive fish assemblages, referring to diversity, abundance, and biomass in the Mediterranean, are along rocky coasts between 0 and 20 meters. However, Posidonia oceanica, as well as coralligenous reefs, are also known as fisheries hotspots for the feeding, recruitment, and nursery sites that they provide to many Mediterranean fish species. Varying techniques have been applied in the past to monitor fish assemblages, commonly depending on the research question or the species of interest. Those may include fisheries surveys, experimental fishing, environmental DNA, underwater video and AI, which is pretty awesome. And one of the most common is UVC, or underwater visual census. UVC are non-invasive sampling techniques used to estimate abundance, size, structure, and fish biomass, as well as quantify species richness. One UVC derivative was developed by Patrice Foncourt in 1999, FAST, in order to take account a predefined list of species targeted by small-scale and recreational fisheries. The technique was originally designed for its simplistic data acquisition and free-range nature. And similar to UVC methodologies or traditional UVC methodologies, it's used to describe the structure and composition of fish assemblages. To my knowledge, FAST has been applied in Corsica along continental French coastlines. It's been adapted to communities, fish communities along the Adriatic coast in Croatia, and most recently it has been applied in Tunisia. FAST has also been referred to as a valuable tool to efficiently monitor fish assemblages in understudied areas, especially through the help of citizen science or research conducted in conjunction with the general public. And this idea is what really sparked the creation of Nacho Dive, a Cannes-based association created in 2017, where I spent my last 6 months. During my time with Nacho Dive, I had the opportunity to take place in 2 6-year monitoring programs, FAST for monitoring fish assemblages, and Seascape for evaluating the integrity of their habitat. Today, I'm going to mainly talk about FAST, but I would be happy to refer to Seascape following the presentation if you have any questions. The overarching goal of my thesis was to fill the need for effective monitoring programs in MPAs and inform regional conservation policy, in this case, through the use of citizen science. With that said, I have set out 3 research questions for us today. Number 1, is fish assemblage health spatially dependent in our study region? Number 2, do no-take zones have a positive impact on fish assemblages? And number 3, does the implementation of no-take zones have family or species-specific benefits? Data in this research was collected between 2018 and 2023, from which I had a direct opportunity to contribute to last year in 2023. Our efforts were focused on 26 different dive sites situated off the coastlines of Esterel, mainland Cannes, the Lérins Islands, Golfe Juan, Antibes, and Nice. Inside the study region are 3 no-take zones: the Capu, Golfe Juan, and Pequot no-take zones seen here in figure A, B, and C. Remember, recreational and professional fishing is strictly prohibited inside these zones. So coming back to FAST. FAST is a UVC derivative based on 6 15-minute replicate counts carried out between 5 and 25 meters depth while scuba diving along a random pathway, as seen here. It takes account the presence, absence, and 2 size classifications of 23 main and 6 rare fish species belonging to 12 fish families targeted by both small-scale and recreational fisheries. Prior to the protocol's execution, citizen scientists were trained to recognize the species of interest as well as identify their size classifications. Following data collection, a base index was calculated in consideration of the number of species observed, their size classifications, and the characteristics of species with high commercial or ecological importance. This base index was then used to calculate the 5 main FAST indices. These indices are used to describe the structure and composition of fish assemblages and include the mean and cumulative indices, the relative species richness, the proportion of large, and the proportion of carnivorous individuals within a given site. Based on these 5 main indices, a cluster health was then assigned from 1 to 5, excellent to very poor, for each sampling site. In addition, univariate analyses, or univariate statistics rather, were used to understand the significance of our results and respond to the research questions that I have outlined today. So this massive effort by 124 observers corresponded to 1,560 15-minute replicate counts and 577 hours in the water, in consideration of a 60-minute dive time. Looking at the assigned cluster health analysis based on location over the 6-year study period, I will first highlight sites with good or excellent health here in Esterel, Cannes, and Golfe Juan, despite a mucilage event noted here in 2023. I will also highlight sites here with average or poor health situated inside the Lérins Islands. Now let's look at these assumptions univariately using pairwise done tests. So here the different colored box plots are corresponding to the 6 geographically separated locations, which I've been mainly referring to. In this case, the mean and cumulative indices, proxies used for species richness and size class occurrence. We noticed that these are, I guess these are great. Yeah. So basically this shows us that Lérins is different than most of the sampling locations that we have observed. We can also look at the proportion of large individuals, which shows us that Lérins has a lesser proportion of large individuals than the rest of the sampling locations. In this case, Lérins was characterized by smaller fish. Further, we can look at the proportion, of large and carnivorous individuals in Esterel, which observed here in light green. Were greater than many of the other sampling locations, but likely because 3 out of 4 of those sites were situated inside no-take zones, and we're going to get into that. So in response to the second research question, Purple box plots represent sites situated inside no-take zones, while gray plots are actually representing sites under fishing pressure. With that said, univariate Mann-Whitney tests in this case indicated significantly greater cumulative and mean indices inside no-take zones. We also observed a greater proportion of carnivorous and large individuals inside no-take zones when compared to sites with fishing pressure. These results begin to explain the positive impact that no-take zone status can have on the structure and composition of fish assemblages. In response to the third research question, I focused on three of the most important fish families inhabiting Mediterranean coastal zones: Labridae, Sparidae, and Serranidae. Here, sites inside no-take zones, once again, are in purple box plots. Small size classes are observed on the left, while large size classes are observed on the right. With that said, we observed no significant difference in Labridae presence. Sparidae individuals for both size classes were more present outside of no-take zones, while on the contrary, both size classes of Serranidae individuals were more present inside no-take zones. This result begins to suggest trophic level imbalance in zones with fishing pressure. We also looked at three species listed as vulnerable by the IUCN, Didontex, Emarginetus, and Cyaena umbra. For all three species, for both size classes, were more present inside no-take zones where we have absent of fishing pressure. However, these strong trends were not observed outside of no-take zones. So what do our results suggest? Well, coming back to the first cluster health analysis, fast observations confirmed locational variation in fish assemblages inside our study region. Notably, sites in the Larens Islands were characterized by low fish densities and smaller fish. The combined efforts by recreational and professional fishing known to frequent the islands, as well as some of the other immense anthropogenic pressures, such as habitat degradation observed there, are likely contributing to this poor fish assemblage health. And in my opinion, this really highlights an area of conservation priority. In regards to no-take zone status, it is known that protections provided by MPAs have positive benefits on fish assemblages. In this study, we observed greater CI, MI, CP, and LP fast indices in areas with absent of fishing pressure. With that said, sites inside no-take zones were characterized by high fish densities, larger fish, and a greater proportion of carnivorous individuals. These high LP and CP indices are likely indicating that large carnivorous individuals occupying higher trophic levels are having a greater response to no-take zone status, likely because these large predators are often the ones that are targeted by fisheries. Despite this clear trend inside no-take zones in our study region, protection can also have positive effects to surrounding fish assemblages via what is commonly known as the spillover effect. This insight may help explain the locational variation that we observed at sites with good health standing situated outside the Kepru and Gulf Juan no-take zones. Looking at family-specific benefits, our results also indicated that macrocarnivorous Serranidae individuals were more present in zones absent of fishing pressure, while Sparidae individuals, mesocarnivorous Sparidae individuals rather, were more present outside of no-take zones for both size classes. In one study, increased Labridae abundance was observed in areas with heavy fishing activity, likely due to decreased top-down pressure by macrocarnivorous Serranidae individuals. To some extent, we expect to see a decrease in macrocarnivorous individuals, or we expect to see an increase in mesocarnivorous individuals with a decrease in macrocarnivorous individuals, such as Serranidae. And with that decreased top-down pressure on mesocarnivores, we then therefore expect to potentially see the decrease in macroinvertebrate assemblages. Therefore, this really highlights an area for further research in assessing prey consumption rates by Mediterranean mesocarnivores in areas with fishing pressure. So looking at species-specific responses, it's clear that we showed positive benefits to vulnerable fish populations, similarly to what has been shown in other marine protected areas in Scandola, Pocol, Banyuls, Metis Islands, etc. Yet we did not observe these strong trends outside of no-take zones. For Imaginatus and Cyaena umbra, this potentially indicates the continuation of illegal fishing, or that we have yet to see actually a full recovery of this species outside of no-take zones. While for Didontics, this really highlights an area of conservation priority, potentially indicating that we need to put in management policy temporarily banning recreational or small-scale fisheries to reestablish their populations. Lastly, although we found the FAST protocol incredibly rewarding, as seen previously in many other studies to monitor fish assemblages, we consider future adaptations to the protocol. Number one, by incorporating numerical counts, this will allow us for biomass calculations for vulnerable fish populations. Number two, to consider including the absence of exotic species, such as the marbled spinefoot or the rabbitfish, which is incredibly invasive in the Eastern Mediterranean. And also, it may be reasonable to incorporate a fixed pathway rather than a random pathway in order to increase the comparability between our survey replicates. Just to conclude, I want to come back to the research questions I outlined today. Poor fish assemblage health was observed in the Larens Islands, really highlighting an area of conservation priority. When looking at no-take zones were characterized by high fish densities, larger fish, and a greater proportion of carnivorous individuals. They also had particular benefits to Serranidae fishes, as well as vulnerable fish populations, Cyaena umbra, Imaginatus, and Didontics. Our results ultimately suggest that FAST is robust enough to meet the same conclusions as scientists, inform regional conservation policy, and fill the need for effective monitoring programs in MPAs and understudied areas. Thank you.
Congratulations, Isaac. Let's go for the questions now. Well, let's start with you, Anne, this time, and we'll go the other way around.
Okay. Thank you for the presentation. Really interesting. My first question is that the non-take zones seem really small. Do you think, in your opinion, that this size is sufficient for a fish population to evolve well, especially after maybe after years of fishing and yeah, do you think it's sufficient?
I will particularly highlight Imaginatus and Cyaena umbra here, because they have a home range of about 1 kilometer. And so if you're able to establish your no-take zones centered around potentially that, for example, for vulnerable fish populations, I think, yes, the size is sufficient. But if it's smaller than that, then no. I think it depends on your goal. It depends on the goal of, yeah.
And you mentioned briefly the IA techniques for monitoring.
Yeah.
What is it about?
The artificial intelligence techniques. Yeah. I know that's being developed both at here at EcoSeas and in the University of Montpellier. Basically, they're setting up underwater video and they're identifying the fish. And my understanding, it's super cool because it sends the identification of the fish right to the Excel document. You just run their analyses and it's pretty much it's cut and dry, you know.
Okay. You think it's better this way than a diver go there and count the fish?
Well, I see that's the thing. And our methodology is very cost effective. While citizen science in general is a more cost effective technique in general. And I don't know what it costs in order to set up these underwater video and continuously run that. So I think there's a trade-off. Mm-hmm. Depends on many things. Yeah.
Okay, and there were 1 or 2 species or a species family where they were more present outside of the non-take zone. Why that? Because of the presence of smaller fishes, or?
So, right here?
Yes.
So I grouped very generally based on family. And very generally, we can consider the Sphaeridae individuals as mesocarnivores. And so what we're showing here is somewhat of a trophic cascade, where if you have less predators, you have more mesocarnivores. And so if you have more mesocarnivores, then in the end, like the common example is the wolf eating, being taken out of the environment, and you have more deer feeding on the plants, and the degradation of the ecosystem. And so I think that's what we're beginning to explain here.
Thank you.
Thanks. I was wondering, how did you train the observers?
The observers go through a pretty vigorous, program. So first they start with a 2-hour theoretical portion before they actually are able to go to the field. Once they make it to the field, then they go through several training sessions underwater to ensure that the data that they're actually recording will go into the final data set to help limit some of that variation that we see beforehand.
And then, so those divers, then they go with different diving clubs and they report everything in one place, or is it only people who dive with natural dive, or how does it work?
Well, I think that's part of the idea, is that once you have these trained individuals, you can they can go wherever, as long as they have the plaquette, and then they can report on what they're seeing. But at the same time, once again, it's you have to have a careful eye when you're not actually with the observers. Once it comes to data analysis. So, but yes, then we also go with natural dive. Yes.
Okay. And then, so in the same line, like when you talk about like to improve the FAST method, you could use like fixed pathways. How would you do that then if people go to do their own diving somewhere? Like, how do you determine you have to go there, follow this line? And
yes, that's a difficult one, because there's actually a study that was done using FAST at Starezo in Corsica. They use FAST to monitor one of their, I believe it's inside MPA, could be wrong. Anyway, they use a fixed pathway in order to replicate their results, and they show it as actually proven to limit some of that variation. But this is also being done by researchers and not actually by citizen scientists. So that's the difficult part, trying to make it more scientific also limits the ability for citizen science to actually engage in conservation initiatives.
Okay.
Yeah, it's a challenge.
How can you make it more scientific? Using FAST method and another method, or how can you do that to make it more?
Something I believe would complement our own presence absence would be the incorporation of environmental DNA in coastal zones. It would provide a more comprehensive outlook on fish assemblages, and especially for some of those elusive species that potentially we might have they may avoid us via their behaviors. So yeah, I think incorporating the DNA.
How does it work? Because I don't know.
No, from what I understand, you take your sample, right? You're either sending it to a lab or doing it yourself, but then once you have the sample, you're able to target specific code in the gene in order to, so if I want to look at, for example, sharks, I'm going to target a specific marker in order to actually understand that what I'm observing is a shark rather than a crocodile. I mean,
So it's quite long compared to.
I believe, yes. I think there's long wait times, is what I've heard.
Maybe I'm mistaken, but you have some results that shows that you have good results, but not in no-take zones.
This one, right there?
No, for the cluster, I think page 10.
Yes.
stable.
Yeah. Yes. Referring to the mucilage event.
Yeah.
So let me flip to the index. Okay, so mucilage is it comes from either I flipped the wrong way. It comes from either how does it? It's a planktic microalgae, or it can be a benthic algae, and it creates these. Better if I show you. But what happens? It ends up swallowing. I can use for lack of a better term, swallowing a lot of invertebrate assemblages. And in doing so, it creates this, here we go. So it creates this very filamentous algae type byproduct from the macroalgae. And it makes, this might be particularly useful for potential like Diplois puntazo, who likes to eat on macroalgae and macroalgae. But for most fish species, it creates an environment, especially for the macroinvertebrates, it's unlivable. Just swallows them whole.
Yeah.
Okay. No. Swallows. Yeah.
Goals.
So good presentation. For the MPA side, I'm not familiar with this area that much. I only just came in. The no-take zones, how well, controlled are they? I know that the previous presentation in the Philippines, they are actually quite well patrolled. So that's my kind of first question. What is the kind of seriousness of this? And also, right at the start, I think you mentioned climate change as well. So how does climate change impact these no-take zones? Do they have any buffer effect? Or are we actually just protecting certain areas and letting others degrade? And that's why we see this difference.
That's a difficult second question. I'm going to start with the first one. No-take zones are typically created around the idea of no extractive activities. In this case, the 3 no-take zones were created by the Prud'homme du Pêche, or the fishing communals, in order to reboost their fish populations, which they all agreed to. So in terms of, how would I say this, management by the, how to say this, the Maritime Gendarmerie, the police, the Maritime Police, rather, it's not necessarily as controlled as I would imagine. However, the fishermen are very good at telling if they see another fisherman based on jealousy of going in that no-take zone and actually removing fish. So to some extent, it's controlled, solely controlled by the fishermen, but also, yes, there's also still a maritime police surrounding that. Yeah.
And for the degraded site, are we just leaving it? Do you think
What? Sorry.
we should always see a difference between a protected area and a degraded site? So how we address this? How can we make sure the environment is, coming up together in terms of recovery, in terms of resilience?
Can you rephrase the question? I'm not sure I understand what
we have some protected areas, and then we have some that are not. So are we not just pushing the degradation to the non-protected area?
Yes, sure. So you're saying how do we ensure that we're not just pushing them to other areas? Yeah. Well, that's the difficult part. I think that comes down to education. Not teaching fishermen, for example, to deploy their nets or to give them the means to deploy their nets in a sustainable manner. And then in that case, we're not pushing the degradation elsewhere. We're reestablishing populations here and giving them the means, once they have the correct gear to use, etc., to sustain the environment long term.
I would have a look at some indigenous techniques from French Polynesia. Dynamic MPAs, a really, really good idea, where we don't just put resources into set area. But also some examples from Japan, Taiwan as well, on natural recovery without any protection, and how they do that. It's a kind of no-take zone as well, but there's not so much resources put into it.
So there's a coming together of people saying, listen, we're going to
what's it called? Traditional ecological knowledge tech.
Yes, okay, yes, of course.
Some of those techniques definitely we could benefit.
Yeah, definitely.
I'm interested in Laren's Island. Yeah. Because on the cluster it looked pretty good. Not the Larens, but the rest of the sites. Why in particular do you think this site is degraded?
You're talking, let me click back.
Page 10, yeah.
There you go. You're referring to here. Why do I think? I think it's a combination of things. I think, well, if you said you're new to the region.
Pretty new, yeah.
Yeah. Well, if you go to the Larens Islands during summer, it's very the boat traffic is very high, and with that comes habitat degradation. Regardless if we have mooring zones to do so responsibly, you still have Posidonia being ripped up by anchors. That's one case, but you also still have fishermen, both recreational and professional. I think there's 30 of them that are from the Campo de Midupêche that are targeting the islands, and in doing so, they're actually still degrading the coralligenous reefs, which are located between 25 to 50 or 60, even deeper meters, and with that comes less fish. So it's not just it's the idea that you're destroying the habitat as well in the area, which is why I think it highlights an area for conservation priority to say, listen, like how much anthropogenic pressure is enough? Until it's destroyed, there's no fish. So,
Thank you.
thank you.
We may have time for last question. For Steve for the sleep, do you want to, before you take your nap, you want to?
No, I have a question.
You have?
Now, you just mentioned in your introduction that 8% of the species were endangered in the Med Sea. How does that compare to the rest of the world? Is that higher, lower?
Higher or lower than the rest of the world. You know, I think a large percentage of that is not referring to, it's more referring to skates, sharks, and rays. Yeah. So in terms of greater than the rest of the world, that's difficult because it's very, it depends on the species you're looking at. So it's difficult to say. I actually don't have the knowledge on it.
Neither.
It's good to
I
say.
apologize. I don't have the knowledge, so I can't speak to it.
Okay, so thank you, Isaac. Congratulations again. Before you leave, you can also watch on the screen. Thank you also to the year one students who are still in internship a little bit everywhere in the world, and Katerina, Nicole, Fabienne, Margos are saying also hello to you. Thank you also to all the speakers since this morning, because you were all on time. It allows us to be right on time and to have 30 minutes break now to enjoy also having discussions in science and not in science. Coming back here with Romane for 3:15. On attend, je vais te présenter, je vais te dire bonjour et tu fais un rebonjour pour être sûr. Okay, so we are back, we're live. So everyone is seated, and so we're ready for you. When you were at CSEL, CESIL, I don't know how you prefer to pronounce it. And so you're going to talk about the comparison of specific composition of marine communities between blue flag beaches of Cannes and Sainte Marguerite Island. Again, we are going to talk about Cannes.
A little bit, yeah.
A little bit, yes.
The subject I changed.
Okay.
The abstract.
That's a good one here.
That's a good one.
Okay, perfect. Okay, go ahead.
Okay. So good afternoon, everyone. Today, I'm glad to present you the work I've done during my internship. So I spent the last few months at the Scientific Council of Larens Island working on a biodiversity inventory, and I was supervised by Stéphanie Pierre. Marine biodiversity is crucial for maintaining healthy and resilient ecosystems. It plays a vital role in ecosystem services such as carbon sequestration, climate regulation. Understanding local marine biodiversity is essential for developing effective conservation strategies and ensuring the sustainability of these ecosystems. Among our seas and ocean lies the Mediterranean Sea, which is known for its exceptional biodiversity, hosting many endemic species. The Bay of Cannes, located on the French Riviera, is a region of both economic and ecological importance. It is home to a diverse range of marine habitats, including Posidonia seagrass meadows, rocky reef, and sandy substrates. These habitats support a variety of marine species that are essential for maintaining the ecological balance. The Bay of Cannes is also a very popular tourist destination, welcoming millions of visitors, yachts, cruising boats each year. On the bay, 7 each have been awarded the Pavillon Bleu label. So this label valorizes the municipalities, beaches, and marinas implementing a sustainable tourism development policy. It plays an active role in achieving the SDGs, especially life below water and clean water and sanitation. On a blue flag beach, you have access to sorting bins, sanitary facilities, facilitated access, and many other criteria. One new criteria is now mandatory to keep this certification, and it's to have an inventory of local marine biodiversity. This inventory helps in understanding the biodiversity of the area and is essential for the environmental management of this beach. So then we can ask the question, what is the diversity of the marine species present on the blue flag beaches of the Bay of Cannes, and how does it vary between the different study sites? So our objective is to identify the marine species and compare them between the sites. By understanding the biodiversity of the area, we can better manage and protect the ecosystem, ensuring its resilience and health. So we can assume that the beach on Sainte Marguerite will have a higher diversity than the one on the Boulevard du Midi, and I will explain later. So how did we proceed to get the biodiversity around the site? So we use scuba diving transect. So we divided our analysis into 2 parts. First, an overview of the biosynosis. To do so, we use transect parallel to the beach at isobat of 10 meters and 20 meters. And we navigated with underwater scooters, and we took videos all along. Here on the map, you can see the different transect that we did. So on the boulevard and here on Sainte Marguerite. Then the inventory of the marine biodiversity. So we did several dives. They have been done in May and June. Some of them haven't been done yet and will be in the next few days. So here we had 7 transects perpendicular to the coast, starting from 30 meters depth to the beach. We did 1 day dive and 1 night dive for each transect, and we took photos of the individuals. And for the one we did not success to take photos, we marked them down. So here on the map, you can see the different color corresponding to the different transect. So this is the Boulevard du Midi, which is an area under pressure with anthropogenic activities, fishing, tourist activities, and resulting in sand each year. And here, Sainte Marguerite, which is part of Natura 2000. So now let's have a look on the results and the discussion. So as we don't have all the results yet, I decided to focus on some specific aspects. So here you have the number of species found on each site during the day dive. So here it appears that Fond de Verre seems to be the richer site in terms of number of species, and Rocher de la Boca seems to be the poorest. This could be explained by the biosynosis of the sites. We have much more flora at Fond de Verre than at Rocher de la Boca, so maybe it could serve as shelters for many species. We could expect to have a higher number of species on Sainte Marguerite, the Natura 2000 site, which is not the case here. As you see, it's approximately the same amount of species. But when we look at the species, we see that there is a difference. So we compare those sites with Jacquard Similarity Index, and we found a maximum of 0.44. It was between Saint-Georges in green and Guérit in orange. So knowing that the closest you are to one, the more similar the sites are. So here it's less than 50% similarity between those sites, and it was the highest number. So the sites are really different. This again can be explained by the biocenosis of the sites, which are different, and also the sediments. So this is the results for the day, and we wanted to compare if we find maybe more species during the night. So we compared 4 different sites, and here we can clearly see that there is a higher number of species during the nights. Also, the color represents the different groups. In yellow here, you can see that there is a lot of fish, and again, more fish during the night. Also, there is some groups that are only present during the night. It's the case for the Annelise at Chantier Naval, for example, and we didn't see any during the day. So in total, it was about 226 species found. So I'm not going to talk about all of these species, so I wanted to focus on some of them. So the most seen species, so a lot of fish, and we saw also a lot of Holoturia around the Boulevard du Midi. All these pictures have been taken during the dives by Nature Dive, so all the pictures you will see in the next slides will be from them during the dives. So we also have surprising findings that I wanted to talk about. So first, the long spinefish, which is a species that is usually found between 50 and 600 meters. And in the bay, we found it at 20 meters. So it was really surprising. It's a species that is not well documented, so we don't know a lot about this one. And it's not just one individual that we found, but 8. So it was really interesting. Then we have the round sea pen. So it's actually 2 species of sea pen, so the Veritulum cynomorium and the Cavernillaria. So the second one was already found in Nice, Villeneuve, and Cagnes. And it was the only place in France that they have been observed. So now we have a new location. So in Cannes, and there is a paper that will be published. So then we also, so this is the second species. Then we have the grease sea pen, which is a species that is found especially during the night because it disappears during the day. We had a lot of observation. It seems to be close to the Veritulum, so maybe they're living on the same sediments. And the last one I wanted to talk about is the Dactylopteris volitens. So this is a species that is rare. It's found in the Mediterranean, but it's rare, and we had a lot of observation along the Boulevard du Midi again. So this was the most interesting species, but there is a lot among the 226. And if you notice, it's only on the Boulevard du Midi that we found them. So we saw earlier that it's important to have night and day dives, but is one dive sufficient to get the more exhaustive list of the species? So we wanted to test it. So let's focus first on the first graph. So we did 3 dives on one site at 3 different dates. So we can clearly see that there is a difference in the number of species we found, with much more species on the 11th. And then when we look at the Venn diagram, we study the distribution of the species, and we see the importance of doing several dives at one site. Why? Because here, between the 3 dives, we only saw 24 species in common, so common to the 3 sites. So on the 11th, we found 22 species that we didn't found on the 9th and on the 5th. Again, 7 species that we did find on the 5th and not on the 11th or on the 9th. So there is an interest of doing several dives on the same site. Here, we did only one dive at each site. Sorry, it was a question of budget restriction for the project. But if we want to get the more exhaustive list, of course, we should do more dives. So now I wanted to talk about the biocenosis results, and the one that I found interesting was the Alophila stipulacea. So this is a species, so it's an exotic species that can be invasive. It comes from the Red Sea, and the species have been found in 2021 by Romane close to the harbor here, with some clear anchoring marks, so we can witness the expansion by maritime traffic. But it has been found on the Boulevard du Midi for the first time in 2024 during the dives. So here there is a clear need to update the map, so it will be interesting. But what can also be interesting is not just update the map, but also to study more in details species, because it can maybe serve as a carbon sink. Why? Because here on the picture, you can see that it captures a lot of organic matter, so woods, leaves, shells, and there is some studies that's shown that it can actually serve as a carbon sink. It depends, of course, of the area, and sometimes it has similar rates than Posidonia meadow. So this could be linked to the small leaves and the surface. But this is something that should be maybe studied a little bit more. So that's what we found in general. So of course there is not all the results as the dive are not all done yet. So what can we conclude now about this finding? So we saw that Boulevard du Midi is a really rich site compared to Sainte Marguerite. We could expect the opposite because it's a Natura 2000 site, so we could think that it's maybe better protected. But we found that Boulevard du Midi is maybe richer. It's an area which is under pressure, so we did not think that it will be this amount of species, and also this kind of species, for example, the long spine spinefish. So we could ask what is really the protection of Natura 2000, and maybe it will be a good idea to maybe expand the Natura 2000 zone. And also we are waiting for DNA results. So we did the sampling in April, and it takes a long time to have the results. So we're still waiting for the results and the last dive. So maybe we will find more species, more interesting species, especially on Sainte Marguerite, because we did not see a lot of interesting species, but we still have the night dives. And we saw earlier that during the night you have more species. So that will be the conclusion. So the Blue Flags Beach are really rich, especially on the Boulevard du Midi. And yeah, we still need to continue the works. So I would like to thank Stéphanie and Damien for accompanying me during this internship, and also a big thank you to Nature Dive team for their work and the amazing picture they took.
Start this time.
If you want.
You want? Allez, let's go on.
Thank you for the very nice presentation. So I understand that your subject has changed, and I can't wait to see the results of eDNA, so don't hesitate to share them. How do you explain the presence of such rare species at these depths? Did you
On the boulevard.
Sorry?
On the Boulevard du Midi.
The little...
That's the thing, we don't know. There were several dives. So they first saw it one time, and they came back after because it was really surprising for the divers to see it, because it's a species that, as I said, lives really deeper. We don't know, I know that close to it there is like a canyon, so maybe it could be linked, but we really don't know why there is the species. That's why we need maybe to observe the population to do some study on it.
Have you checked in the literature?
No, not necessary, because it's like you have in the literature little information. You have one, usually it's just to describe the species, and you have one paper that's studied them, but just it doesn't say anything because it's supposed to live deeper. So in the literature they found it between 100 and 200 meters, but I didn't see in any paper less deep. So I don't know.
Do you know the Doris website? Okay, because there you can add your observations. Maybe you did that.
I personally didn't did it first, but yeah, it will be interesting to put maybe some other people. But even in Doris, they said it starts from 25. Sometimes you can see at 25, but usually it's between 50 and 50 and 600 meters, and it usually live between 100 and 200.
Maybe it can be updated now with your results.
Yeah, of course.
You said that the inventory of the local biodiversity was new. Since when exactly? In the
criteria?
Yeah, the criteria.
So the criteria, I think it was for 2023, and as it takes time, it's to validate the criteria, you need to either have your inventory or it's in process. So
now
that's why it's validated, because it's in process.
Okay. And how many persons were there to observe, to take all the data?
For each dive, you had 2 people, 2 divers, only in the water, and 1 on the boat.
I hope they had good eyes.
Yeah, and they're professional, so they know also the species, because for the one you cannot take a picture because sometimes it just go away. It's important to have people that know the species. Yeah.
The difference with the environmental DNA. But it's another debate. Okay, thank you.
Okay, thank you. I was wondering, when you show the differences in number of species per site, did you try to look at the substrate too? I suppose it's different depending on, if you have Posidonia, if you have a rocky reef in front of the beach, it will probably impact the results.
So I have the data for the flora we had. So we did the biosynthesis. So for the biosynthesis, it's a video, but it's a video, but parallel to the coast. So as the biosynthesis can be one thing here, but we did the transect like this, so it doesn't give really the biosynthesis at the site. But what we have is the depth of the picture. So when the picture is taken, we know at what depth it is. And usually we have photos. It's hard to see. Yeah, it's like semi-substrate, but no, we don't have exactly we know the biosynthesis in general. So on the transect, we found Alophila, Posidonia, blah, 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 but we don't know exactly where.
Okay, because this could, of course, really impact what you see. And so one thing that was not clear for me, and I'm starting to get it, it's like when you say number of species, it's everything that are like, plant algae, everything was in.
It's all of these groups.
Okay. And then, if you stay on that slide, so how do you explain that things that don't move are not the same between day and night?
Because I expected this question. It's not the same transect, so it's around the same area, but it's not the same transect. So if you do your transect here and then the transect at night, you do it 10 meters apart, maybe that's why we don't see it. But yeah, that was surprising to have like porifera that's not supposed to move and they're not here. So it depends, it's more an area. That's why it's important to combine both because you don't have the exact same transect, but it's really close.
Okay, so that was the same idea when you compare the 3 different transects. They were also in the same area, but like, or not like,
Yeah, that's
transect that were.
Yeah, we try to stay around the same area.
Yeah.
But of course, it can differ a little bit because when you dive, you don't know exactly where you are.
Yeah, I mean, it depends the kind of transect you do. If you put like
No, we
lines in between, you can do it.
from a point, and then we mark the GPS coordinates, but underwater, you don't have the coordinates, so you don't know if you see exactly. And here, the goal wasn't to compare really. I just decided to do it because they came back many times because they wanted to explore more this area because it was surprising area. So the goal wasn't to really doing replicates. So that's why it's not exactly the same, but still it's in the same area. As a beach is really long, and we're doing like a perpendicular transect. So
Thanks.
Thank you, Steve. Nadia, we will finish with Aziz.
For the Jacquard Similarity Index, can you explain a little bit? Yes, this one.
So the similarity, the Jacquard is supposed to be a dissimilarity index, and I transform it in a similarity index so it's more understandable. And so we compare the different species. So I had like I had data with all the sites and all the species. And then we mark down like 1 for presence and 0 for absence because it's only qualitative data. And so at the end, they compare all the sites. So if they have the same species between the sites, and this was the matrix. So this was the higher number we got, which is not really high, honestly. So the sites are completely different.
Don't have any.
Cool. I think it's really good to include a night dive in your study because I've done a lot and you can clearly see a huge difference. Why do you think this difference is?
I would say because there is, first of all, less activity, so less anthropogenic activities, boats. Also, their fish can hide from predators. I would say this, and these are also nocturnal species, such as the pteroids that we saw. So it's interesting to have, of course, night dive.
And you mentioned that you don't have this kind of baseline character of the reef data. You didn't do a kind of concept to just get the character of the reef.
No, we did not. No. We did only. So the biosynthesis was only to get like an overview of the biosynthesis in general, but we cannot compare with the species we found because it was parallel transect. So it's like a small parts that we can compare. And yeah, no, we don't have the reef character.
Because it'd be quite interesting to look at things like structural complexity and things like that. Is obviously from the bay to the island, it should be very, very different.
Yeah, definitely.
And the night dives were not done on the same day as the day time.
No, it was a different day, but it was between late May and early June. So it was kind of the same period. It wasn't like a month apart, but yeah. It wasn't on the same day. It was a different day.
And just one last comment on your presentation. I think a good added thing you could do is relate the CO2 capture back to, in your introduction, the resilience part, make it more obvious. Really kind of.
Sure. Yeah, because I was talking about carbon sequestration in general, but not necessarily this one. Yeah.
Also, the eDNA would be good as well.
Yes. But eDNA, I didn't talk about eDNA because I have like nothing on it. That was the idea of the project was first to compare the two methods to see if maybe we can just replace the dives between the dives and just keep the eDNA, which I think it would be better to combine both eDNA and dive, but we didn't get the results in time, and I'm not even sure to have them for the report, so that's why I didn't really talk about, just like in the conclusion, the perspectives. So that's what but it's interesting, and it's very frustrating to not have the data for eDNA, because I think it would be really interesting to see if they actually capture the same amount of species.
Final question, what light did you use in the night dive?
What
Type of light?
I have personal no idea. It's nature dive. That's the dive. So we can maybe ask Samuel what kind of lights.
Sorry.
Yeah, but I don't know. I don't know. They took photos of it, and yeah, maybe for disrupting the
We do have about time for additional questions, so I refer to Steve as usual, who has to understand.
Yeah, I have a question. So you mentioned the carbon storage, and of course. So how can you tell that? What is the definition of when can we say that the coastal ecosystem is a carbon sink?
What I read, it's they accumulate carbon storage in the sediment, and it's the case for Posidonia. So here, there is one study that said that it's not true. It's the sediment that really capture the CO2, and the leaves help. Well, the species helps to capture it, so it captures a lot of organic matter. And then there is carbon accumulation in the sediment, and it's actually what they say. It's the fact that it's a simple architecture with small leaves and a small area between like the sediment and the water, so that helps to store carbon. But it depends on the area. So in some area. It will not capture carbon at all. So that's why it's important to study on the Boulevard du Midi to see if here it actually sequesters carbon.
Yeah, I think that's the important part is it sequesters carbon in the sediment for at least 100 years. So, since those algae are new there, it's hard to tell for now. If you take like a core in Posidonia, you can tell, okay, it's very old carbon, but here.
Yeah, and on the map, we didn't know that there was allophila. It's really since the dives that we now know that there is some allophila, so maybe we'll be interested.
Potentially, yeah.
So for now, it's not invasive. There is a lot, but it seems to be not invasive. It's exotic, but maybe it can serve the ecosystem services. Maybe if we found that finally it has a high rates of carbon sequestration. But it has to be studied.
All good. Anybody wants to ask a last question? All good. Thank you, Roman. Yeah. And also thank you guys online for supporting Roman. You will see the comments and the uploads. So we are now going to the last presentation of the day with Emma, who is listening to us from a distance. Because Romane, her life she has is actually in Costa Rica. So that's her in. Do you hear me, Emma? You're mute.
Now, can you hear me?
Voila. Yes. We're all here for you. You can see us. Yes. So you're already sharing your screen. I will just check if it's working well. Yes. Before we start, I'd like to ask that to all the students being online with the distance. What time is it in Costa Rica?
7:47 of the morning.
what? Of the morning. Okay.
Yeah.
So I guess you're ready to go. I'm just asking you the question: do you prefer to be alone on the screen, or do you like to see people at the same time when you present?
Oh,
Yes.
so
Like that,
I
or
don't,
just alone?
no, with people is fine.
With people is fine. Okay.
Yeah.
Allez.
Like this is, yeah. Thank you.
So you
for
have the
asking.
next 15 minutes. Go ahead.
Okay. Thank you very much. So hello, everyone. Good morning or good afternoon. I'm sorry I couldn't be on site to present, but I'm really happy to share with you the results of my master thesis on the restoration of the Ricardo Reef in Bahia Culebra in Costa Rica. It was a comprehensive approach to coral transplantation and ecosystem succession. I had the great opportunity to do this thesis at the Universidad of Costa Rica at the Laboratory de CIMAR under the supervision of Juan José Alvarado. So this is a little overview of the agenda of today, and I'll start by introducing you to some context on coral reef. As you may know, coral reef are a very diverse ecosystem. They are home to a quarter of the ocean species. Unfortunately, they are facing environmental and anthropogenic threat, which led to the loss of 80% of the coral cover in the last 30 years. Today, coral cover less than 1% of the ocean floor. The impact of these threats include coral bleaching, with a reduction of coral cover and a loss of structural complexity. This eventually leads to the loss of biodiversity, a decline in biodiversity, notably in reef fish species, as observed in Bahia Culebra, northern Pacific of Costa Rica, with a decline of 49% of reef fish species between 1990 and 2015. The Eastern Tropical Pacific, also referred to as the ETP, is particularly concerned by this loss in coral cover. In Bahia Culebra, so northern Pacific of Costa Rica, as you can see on the map, have known a dramatic decline, going from 38.5% of coral covering the bay in 2006 to 1.1% in 2022. The main cause for this decline is the El Niño Southern Oscillation, the ENSO. On this map from August 2023, we can see the sea surface temperature anomaly reaching up to 2 degrees Celsius for a too long period of time above the threshold of temperature for healthy coral growth. This high temperature produces bleaching and lead to the death and the loss of the corals. And this is what we observed in the Ricardo Reef, where we developed our experiments. The first picture was taken in August 2023. So this is the reference site. We can see a healthy reef with the first sign of bleaching. And 2 months later, October 2023, we can see the reef all bleached because of the high temperature. And sadly, we can see that months later, the reef was all dead as it did not survive the high temperature for a prolonged period of time. And this is our control site. So the aim of this study is to restore and help in the succession of Ricardo by introducing Poecilopora fragments from 3 different locations within the bay. So for this, we look at the restoration method of transplantation as we observe promising previous results in other parts of the Eastern Tropical Pacific, notably in the Mexican Pacific and in Colombia, with high survivorship higher than 89% for both studies. However, it's important to keep in mind that the transplantation faced some challenges, notably the benching competition. And now the star of this project, the species Poecilopora, which is a coral species the most abundant in the Bahia Culebra, that is resilient to a variable environmental condition and has a rapid growth of 5 centimeters per year. So for each objective, we formulated an hypothesis. So the first hypothesis stated that implanted coral fragments hold a substantial capability to grow and replace the lost reef. The second hypothesis stated that living coral fragments show significantly diverse levels of success in contributing to the overall growth of the reef according to their original location. And last but not least, the third hypothesis stated that the growing new coral significantly enhances the fish biomass compared to the lifeless reef. Now, let's go over the material and method employed for the study. So as I mentioned, the study took place in Bahia Culebra, which is located in northern Pacific of Costa Rica in the region of Guanacaste. Bahia Culebra is a sheltered bay with a maximum depth of 42 meters and an area of 24 kilometers square. As all the country of Costa Rica, it is subject to 2 different seasons. The dry season going from December to April, and the rainy season going from May to November. The tiny yellow dot you can see on the map is the Ricaro Reef, where we developed the restoration project. The Ricaro Reef depth varies between 8 and 10 meters depending on the tide, and it has a very small area of less than 0.1 kilometer square. And the 3 blue dots you can see on the map are the donor colony sites from which we've taken fragments to implement in the Ricaro Reef. We have the reef of Guiri-Guiri, Matapalo, and Zapotiel. Those 3 reefs are different in location within the bay and also in depth. Now that you can position the Ricaro Reef, let's dive underwater to see the detail of the experiment. So for this experiment, we implanted 3 different experimental transects, each hosting the fragments from different locations within the bay. So the first transect hosted the fragments from Guiri-Guiri, the second from Matapalo, and the third from Zapotiel. Each experimental transect has its associated control transect parallel to it, and the area for each transect was 20 meters squared. Now, let's look a little bit closer in the experimental transect. We can see that we implemented 100 coral fragments on each experimental transect. So a total of 300 coral fragments were implanted in the Ricaro Reef. And each fragment were attached to nail with the help of a zip tie, as you can see on the picture on the top right of the slide. Now, continuing our underwater expedition, we monitored the coral cover using underwater camera and a PVC photo quadrant frame. The output picture gives us the picture we have on the top right of the slide that was analyzed through the software ImageJ to measure the area of each coral fragment. Additionally, we monitored the fish biomass. For this, we recorded the fish species, their length, and the number of fishes observed for each experimental and control transect. The study was conducted over 3 monitoring days at day 0, 28, and 70. And we just came back from the field 2 days ago. That was day 119, but sadly, I could not include those results in this presentation. And the fun part of each research project, the statistical analysis that we conducted through RStudio. So we formulated a null and alternative hypothesis for each objective, and to evaluate the potential succession of the disease rift, we used the Fisher test. To compare the success of living coral fragments, we used a Freeman and Wilkinson test, and finally, to investigate the contribution of the new corals to fish biomass, we used the Crystal-Wallis and Wilkinson test. So the first look at the results for the coral cover comparing the experimental against the control transect, we can see that we have a coral cover that is present all along the 70 days for the experimental transect, and for the control transect, as expected, there is no coral cover over the 70 days. Despite this observation, the p-value was 1. There was no significant difference in the success of living coral cover in the experimental transect compared to the control transect. But now let's come back to this graph just a second. We can see that from day 0 to day 70, the coral cover decreased from 4.11% to 1.99%. This decrease can be due to the transplantation stress undergone by the coral fragments during their collection and transportation, which may have inhibited their acclimatization to the restoration site condition. Additionally, the fragments were transplanted directly on the seabed, so in direct competition with the red microalgae Hypnea, which may have contributed to the death of some fragments. Also, the study was conducted at the end of the dry season, and previous studies have exhibited higher growth rate of coral fragments during the rainy season, which might have been a factor as well. And finally, it's important to mention that the study duration was 70 days, which is very short, as most of the restoration studies using transplantation method last at least a year and exhibit significant and positive results after 3 months. Now, let's look at the coral cover comparing the 3 experimental transects. The first look at the results, we can see the global decrease in coral cover for each transect. But looking a bit closer, we can see that from day 0 to day 28, there is a slight increase in coral cover for the transect 1 hosting the fragments from Guiri-Guiri, and that the overall decrease for this transect is of 0.3% loss of coral cover, whereas the loss of coral cover for the 2 other transects is higher than 0.7%. Despite this observation, the p-value was 0.097. There was no significant difference in living coral cover between the transect originating from Guiri, Matapalo, and Zapotiel. But let's come back to this transect one for a second. We can see that even if there is no significant difference, the fragments from transect one coming from Goury seems to be a bit more resilient than the two other transects. And this might be due to ecological memory. It happened that the coral from Goury are coming from a restoration site. So the fragments from Goury have been transplanted in the past, possibly enhancing their resilience to future stress, including the transplantation they have undergone at the beginning of the study. In contrary, the fragments coming from Matapalo and Zapotial are not coming from a restoration site, they are coming from wide reef, which are not subject to restoration project. Also, it's important to mention the short duration of the study, which might have inhibited the significance of the results and give time to coral to readjust to their environment. And last but not least, let's look at the results of the fish biomass comparing the control and experimental transect. At a first look, we can see that the fish biomass for both transects are very close. We have a 97 gram per meters at the control transect and 61 gram per meters for the experimental transect. As expected, the p-value was 0.45. There was no significant difference in fish biomass in the experimental transect compared to the control transect. I would also like to highlight that over the 70 days of experiment, we've observed 25 species, both in the experimental and control transect, but that 6 species were exclusively observed in the experimental transect, while 3 species were exclusively observed in the control transect. So despite the non-significance of the results, we see that observations tend to exhibit a higher fish biomass for the control transect, but a higher richness for the experimental transect. So despite, contrarily to our belief, coral transplants do not always enhance fish colonization, as rocky and rubble reef habitats provide sufficient resources to meet the coral reef fish's need. So this might explain the very close mean and non-significant of the results for both the control and experimental transect as well. And also, here too, the study duration was short, as with a higher fish database that we've collected through a longer period of time, we could have drawn more conclusion from the results. So now, to summarize the importance of this project, it's important to acknowledge the cumulative effect of the ENSO events, which made the ENSO event of last year the fourth global coral bleaching event with a bleaching alert 5 that resulted to the loss of multiple coral reefs. This underlined the urgent need for coral restoration action to preserve this precious ecosystem. And this is why we developed the restoration project on the Ricaro Reef, hoping to extend this effort to other reefs within the Bayac-Foulepas. Sadly, the restoration site of Ricaro did not replicate the condition of the reference site from August 2023, so the restoration project cannot be deemed as successful. Indeed, none of the hypotheses could be accepted as the results were non-significant. But despite the non-significance of the results, we could draw some interesting observations which help us build a future perspective for this research project. Notably, that we will keep monitoring the reef, so extend the study duration to at least until the end of the year, to determine if the observed changes are due to the restoration effort or natural variability. Additionally, we're thinking of implementing sea urchin that are macroalgae grazer capable of controlling macroalgae expansion on the degraded reef and to reduce this macroalgae competition. Also, the study took place at the end of the dry season, and we now entered the rainy season, so we are hoping to see a higher growth rate of the coral fragments, as previous studies exhibited higher growth rates during the rainy season. Also, it will be interesting to explore other intervention methods in the transplantation process. So maybe having larger coral fragments, which could enhance their resilience to the transplantation stress and also the macroalgae competition, and maybe put them a little bit higher on the nail so that they are less in competition with the benthos. Also, we could explore further this idea of ecological memory to determine if taking fragments from the restoration site could improve the efficiency and resilience for future restoration projects. And finally, I would like to mention that in parallel of this project, we developed a photo mosaic of different reefs in the Bayac Culebra that can facilitate the tracking of broader changes across the entire reef landscape. So this is still in development, but this could really help pinpoint where the restoration project would be the most efficient. And finally, as I mentioned previously in the presentation, we came back from the field 2 days ago, and just to give you a little update on the corals, sadly, the corals of Matapalo and Zapotial are keeping a decreasing trend in coral cover, but the corals from Guri are quite healthy. So I would like to thank you very much for your attention today. I'm sorry I couldn't be with you on site, and I would really like to say a big thank you to Juan Jose and all the lab of CIMA, with whom I have had the most amazing experience, and I learned so much. And if you have any question, I'll be more than happy to answer them.
Sorry, Emma, congratulations. You were talking about resilience of corals. I think we can also talk about your resilience. I don't know what's happening around you, but you kept calm during your whole presentation. So congratulations.
Yes, I'm so sorry.
No, we only heard you. It was fantastic. It's not.
I was like, no, what's happening? Okay.
So let's go for the questions. Yes. Allez, vas-y. For you.
Really good presentation. I know how difficult oral transplantation kind of stuff can be and disappointing, and a lot of times it is. So I wasn't really surprised at the results, if I'm honest. My first question is, how do you know they are all the same species?
Oh, so how do I know they are the same species?
Yep.
Right, okay.
From the fragment.
The fragments. So actually, the Pocillopora. So I've simplified it for this presentation, but there are two different species of Pocillopora that I don't remember the name, but they are like two similar species and very hard to distinguish. So we're like joining them into like the same species Pocillopora. So we know that for this project we only took Pocillopora species because, first of all, in the bay, it's like the most abundant species, and they are quite. It's a gender that is quite recognizable. So all the fragments we took, they are coming from reef of only Pocilloporas. I'm trying to find the name of the two species. Yeah, Pocillopora damicornis and Pocillopora elegans. So those are two different distinct species, but super hard to distinguish. So it was a mix of both of those species of Pocillopora.
So the main event which caused them to decline, the initial reef to decline, was the El Nino event, or coral bleaching event.
Yes.
So the reef before the bleaching event was actually, I mean, it's Pocillopora dominated, but it's quite healthy.
Yes, before the bleaching event, the reef of Ricaro was one of the most healthy in the bay and very, very resilient. It actually has been very resilient to previous heat waves the bay has encountered.
So why do we need the restoration? Because we know this reef can recover already.
So that's maybe I can I share the slide, because I had great, thank you. I had the I don't think it's voilà. I think it's no. Sorry. Yeah. So here on the picture from November 2023, you can see the reef was completely dead, and there was no living fragment at all. So there was no possibility of the reef to regenerate. So we wanted to help in its succession by implanting some fragments to see if it was possible to restore the reef and to, of course, in 4 months I wasn't expected to have it back as in August 2023, but we were hoping to start a restoration of the reef as there was no living fragment. The whole reef, it was only rubble. Very, very, very sad. I don't know if
So,
that
yeah, in your results, the coral cover went from 38% to 1.1%.
The global coral cover, I'm not controlling the slide well. The global coral cover for all 3 experimental transects decreased from 4.11% of the coral covering the whole experimental transect to 1.99%. So we actually had a decrease of more than 1% of the coral cover in the bay from the coral we've implemented.
But I remember in your introduction, you mentioned there was a decrease of 38% coverage to 1.1%.
At the beginning, so, I'm so sorry, I could not hear you well. Maybe I can share my slides again. Yes, thank you. I don't know why, but I'm trying to. Well, doesn't want to go before. I want to go to the. Sorry about that. I'm just trying to come back to the slide, but it won't let me for some reason. Sorry. Yes, here. You're talking about here, right?
Yeah.
Yes. Sorry. So yeah, between 2006 to 2022, there have been a really dramatic decline due to El Nino event. But actually, it's important to note also that between 2006 and 2012, there has been a lot of reptile in the bay that have participated in the loss of a lot of reef
Okay.
as well.
I've another question about the fragments themselves. What stage of the mother colony you took them from? What stage was it at? Because they will have different growth rates and depending on different stages. And there's a really good paper from Yossi Loya last year that talks about the kind of reproductive memory. So this idea of ecological memory. The fragments will remember their age rather than size. So some of them may be reproducing, for example, and some of them may not be at reproductive age. So how did you determine what size of colony you were taking from?
Okay, what size of the colony, right? Not the fragments, you mean? Yeah. So to be honest, we did not pinpoint specific colonies as we went down on the reef of Guéry. We took fragments both from the colonies we were restoring. So there were very healthy colonies that exhibited no sign of bleaching and yeah, they were healthy. So I honestly can't tell you how long they have been growing, but I know it was a healthy restoration reef.
Okay, thank you.
Thank you, Aziz. We have time for 1 or 2 more questions. I know if you do have.
I have questions.
Okay, one for Steve, one for Anne.
Yeah, I was a bit, when you look at the fish biomass, were you really expecting to see anything with, 1% coral cover, and how do you think this would impact the fish? Would they really care if there is like 1 coral there and there, or no corals at all?
So to be honest, as I mentioned, the duration of the study was very short. So of course we weren't expecting like a significant difference. Also, with as the coral didn't have time to grow that much. No, but we thought it was interesting to measure the difference, see if the corals could add something to the reef that would bring back some of the reef fishes that were not there anymore after the loss of the reef. So yeah, to answer your question, we after we'll keep monitoring. So this project will keep going this year. We'll keep monitoring both the coral cover and the fish biomass, and to see if we're looking at difference. And we were also I could not include all of that in the presentation, but we were also wanted to look at the difference in community composition. That it's written in my report, but I hadn't had time to include it in the presentation. 15 minutes was too short. But yeah, we were looking at the community composition, and this is an interesting trend we would like to observe in the future if there is a difference between the control and the experimental.
We have time for questions from you and from a special guest.
Oh, special guest. Thank you for the very nice presentation. I really enjoyed it. On the photo of the coral in November 2023, is there mucilage or are there polyps of the coral? On the last photo presented at the beginning, even at the end.
Okay, yes, this one.
Yeah. Is it macroalgae competition?
Yes, so it's those are dead corals that have been taken over by the macroalgae.
Okay. And no.
Yeah. So the special guest is actually, so it's customary to propose the supervisor to also ask a question. I hope you're not surprised, but Juan Jose is asking this question to you. Decision maker tells you that the restoration does not work. So how do you explain to him that his investment in this type of activity should be viewed calmly?
Yes. Yes. So if a decision maker, thank you for your question, Juan Jose. So if a decision maker tells me that the restoration does not work, I would tell him that we only have 70 days, right? 70 days over the course of like all of the El Niño event that happened, all of the time it takes for a habitat to restore is really, really short. And the key is really to be patient, to observe some results. So even though we have observed a decrease in coral cover over the 70 days, it's important to be patient because the coral fragments, they need time to readjust. We transplanted them from their habitat to another. It's like if it's a lot of stress on the coral, it has to readjust. So in between the time we see some positive result and the time it needs to adapt, we need to be patient and put the time and energy in a restoration project to see positive results. I hope it answers your question.
I hope. So he also told you that it was great to have you in Costa Rica. And by the way, also you have some congrats from different people that you might know. Just showing the same family name. And voila. So it was also good to have you even with the distance, Emma. And so it marks the end of so first, congratulations, Emma, again.
Thank you.
And so also a few words for tomorrow and Thursday. So tomorrow we are going to talk about space technology and science and socioeconomics. We're starting, so I talk, of course, under the supervision of Crystal on the back of the room. We're starting at, so the first presentation starts at 9:30. Also under the supervision of Vincent. Do we have breakfast tomorrow morning? Yes, so you're welcome to come from 9. Okay, you have a nice breakfast, and we close the door at 9:30. Voila. So the first presentation of Maya will not be online, but you are welcome to come here to support her. At the end of the day tomorrow, the presentations of Esther and Margot will be fully private. So you will finish your day around 3:30. We'll continue with Esther and Margot on our side. And then the day after, it will be also with, tomorrow afternoon, we also have the consultancy and finance session with Zoe, Alice, and Céline. So I mean, I made a mistake. I'm sorry. I'm sorry again. We finished with Nikita here. We continue with Esther and Margot as a private session, but we did it so that at the same time, you can move in another room and participate, attend to the presentation of Zoe, Alice, and Céline. So if you're nice people and you want to support them, you can stay until 5. Okay. We will re-explain all of that tomorrow, and it will be easier. Well, for Thursday evening, Christa, do you want to say a few words about the activity, or I continue?
Oh yeah, sure. I can share. So as you understand, we did organize some activities for you as team building on Friday. So for sure, what I need is just to be sure that you are going to be present. There will be an activity for 2 hours in the morning from 10 to 12. Then the idea is to share and picnic together at the beach, and then we can finish in the afternoon with a little pétanque, like really French, also. So just let me know if you are present. It's really important for the activity in the morning for sure. But I need to confirm that.
So the activity in the morning is in Antibes?
Yes, all the activities are going to be in Antibes.
Yes, in Antibes, and so it includes some kind of, it's inspired by the Olympics, which are, which are hosted by France.
Spoiler.
No spoiler, but avoid heels, for example.
Yes.
Also, before that, I was also talking about Thursday afternoon. Thursday afternoon, as usual, we like to decompress and go to a pub. Yes, it's MARRES, so you know. We never force people to drink. There is plenty of water, and good thing in France, it's for free. On est live, yes. Yes, welcome to MARRES. We all know that. So on Thursday afternoon, there is, as I say, to decompress and also to watch the video pitch of the year 1 students who are with us. I know not all of them are here. And so to have also nice chats outside of a presentation room with the staff, with the colleagues, with you, future colleagues, because you will work all together. This will be at the Manolans in Nice city center, so Saleya. I'm just saying that because there are 2 Manolans pub in Nice, so it's not in the port. It's the one in the city center in Saleya. We will welcome you there from 4-ish, something like that. 4 and active with really start the activity at 4:30. And so I really hope we will see you. And if you want to bring people with you, it will be really nice just to tell us, to ask us before, okay? But it's possible, just tell us that. I understand that some people from Ecosis are coming and they're welcome. Okay, so thank you for your participation, for being here today. It's really important. See you tomorrow at 9, something like that. Bonne soirée. Merci Emma d'être restée.
Merci beaucoup.
Salut Emma. Au revoir.